Welcome everyone. Thank you for joining us for our third webinar in the Advancing Care Through Genomics virtual speaker series. This is part of our Individualized Medicine Conference virtual experience. This is our third webinar, and this will continue for another one coming up in October. So we hope you continue to join us. Today we're excited to have Dr. Acosta and Dr. Marina Walther-Antonio from Mayo Clinic join us for our discussion on Beyond DNA in Cancer and Obesity. Dr. Acosta is an assistant professor of medicine and gastroenterology at Mayo Clinic. He's a physician scientist trained in gastroenterology and obesity, and his main interest is in the complexity and heterogeneity of human obesity. His research focuses on mechanisms and regulation of appetite, satiety, and understanding phenotypes of obesity and how that relates to response to obesity interventions. Today, his talk will feature data from several studies that look at advancing our understanding of the relationship between obesity phenotypes, biomarker discovery, and pharmacogenomics. Dr. Marina Walther-Antonio is an associate consultant in the Department of Surgery with a joint appointment in the Department of Obstetrics and Gynecology at Mayo Clinic in Rochester. She's an assistant professor in the Microbiome program, and her research focuses on the study of the microbiome in human health and disease, particularly in cancer and reproductive health. So without further ado, we will invite Dr. Acosta to start our discussion today. And one last reminder, if you have questions, we'll leave those towards the end, and please use the Q&A section of the Zoom webinar to enter your questions. Thank you and welcome, Dr. Acosta.
Thank you so much, Dr. Kedis, and everyone at SIM for inviting us to present in this very interesting and new frontier of where we're talking beyond DNA. And we are delighted to share this with Dr. Walther-Antonio and talk about cancer and obesity. So I'm delighted to be here. And as Dr. Kedis mentioned, I'm an assistant professor of medicine in the Division of Gastroenterology and Pathology. A few disclosures of things that I may be talking is a license that has Phenomic Sciences, and this is that myself and Mayo Clinic has a financial conflict of interest in that has been approved by our Conflict of Interest Review Board. So let's jump into the topic. First, I'm going to put the setting of what is obesity. Obesity is a disease that is a chronic disease. It affects 40% of adults in the United States alone. More than 1 billion people in the world is suffering from obesity. And it matters because it leads to heart disease, stroke, type 2 diabetes, cancer, and premature death. It also matters because it costs us $480 billion of direct annual healthcare cost. And if we add the indirect healthcare costs, it's half of our healthcare budget is spent on obesity-related conditions. And it also matters a lot more is because the current existing treatments are mostly ineffective. And as what I mean by this is that most of you have experienced this or talked with someone. We have countless diets, we have devices, we have medications, we have surgery. And unfortunately, they don't work. There's one main reason why these things are not working is that the one size fits all is currently not working. We keep thinking that the next diet is going to help everyone. I'm going to try this diet and this is going to help me. And unfortunately, we have not advanced any further in trying to find out what is going on. And what I mean by this is explained on this graphic, which is quite simple to understand, but let me walk you through it. Here in the y-axis, we have the percentage of patients who are participating in different weight loss interventions. And then we have diet, medications such as liraglutide, devices such as the duodenal liner, and then bariatric surgery such as the gastric bypass. And then down here on the x-axis, we have the percentage of patients who will gain some weight when they get into these randomized placebo-controlled trials, those who lose a little bit of weight, like 0 to 5% of their body weight, and those who lose a lot more of their body weight, for example, lose more than 40 to 50% of their body weight. And doesn't matter what is the intervention, we can see that there's a very heterogeneous response to the interventions. Initially with a low carb diet, we can see the responders and non-responders. Same thing with medications, devices, and surgery. So it doesn't matter what is the intervention, we have good responders and poor responders. And clearly this is one of the most common problems when we try to treat not only obesity, but most of chronic diseases. We just don't know who is going to be the best responder for the intervention. And the new era of precision medicine, we need to change that paradigm. So what do I mean by that? So far in medicine, we have had this traditional approach in which we look at diseases and their symptoms and signs and symptoms, and we call those phenotypes. We label those as a disease. We go through an expensive either diet, drug discovery, device discovery, or even surgical approach. We go through very large and expensive trials, and then we have what we're seeing right now, this highly variable outcome. But precision medicine is bringing a new paradigm. It's bringing a new way of looking into things. We need to look at the disease that is breakdown in phenotypes, and only those phenotypes will have multiple different phenotypes that we can understand the underlying pathophysiology of each phenotype, with what I call here the deep phenotype. Then we can apply omics in way of using our biomarkers, but also to help us understand disease. Use these omics to help us as a companion diagnosis in the approach that we want to take. For example, I'm talking about here drug discovery. Do a more targeted trial, as you can see here, that it will be cheaper and faster to get through, and will have better outcomes. So how will this be looking, and how we can use these tools is the next frontier, the next question that actually brings us beyond DNA. We have learned a lot about DNA, and for example, in cancer, we have a lot of now new markers that are DNA driven that are telling us how to choose the next medication and how to tailor precision medicine approaches for patients in different diseases. But that has been great, and we have advanced a lot the field in the last 2 decades with DNA. But then we need to talk about the rest of the omics. And there is so many omics: proteomics, transcriptomics, genomics, pharmacogenomics, and we can just keep calling omics in any of the studies of any of these things that are new tools to understand disease, as I put in this graphic. So what exactly is a omic? In a very simple way that was reviewed in this review in 2007 by Detmer, he tells us that there's this omics cascade. We have the genome, that is basically what can happen. And I'm going to put it in the most simple terms as possible, as we're going to make a cake. The genome is when we go to our library, we find that book with recipes, and we pull it out, and we open the recipe for that chocolate cake that we want to make. What appears to be happening is when we look at the transcriptome. We open that book, we start reading the recipe, and we start making the recipe. What makes it happen is the proteome. So now I have the cake already, all the ingredients mixed. I'm going to put it in the oven, and then we'll see how it comes. And then what has actually happened, and what's happened is the metabolome, is that cake is being baked in the oven, and eventually when I take it out, I'm going to try it and see if I'm a good chef or a bad chef, and that will be the phenotype when we talk about our cake. The same simple process is used in every single process in life, even though to this point this was used to be called a dogma. Now we have proven this is wrong. This is the simple way to explain it. We can actually complicate it a lot more, and during these talks today, you will be hearing about this, which is actually way more complex than what I just described. We have our genome that still comes from mom and dad, but then the epigenome is regulating our genome. And then we have the transcriptome, and then we have the proteome, the metabolome, and we have some things like the microbiome. And while the genome is very stable and is the same for us, it changes through our life based on other external factors. And then we also have the environment, which is playing an important role in different diseases. So if we have a very simple genetic disease that we know that a gene is mutated and that gene is going to give you a disease, it's very easy to understand the genetic predisposition for a disease. But when we talk about more complex heterogeneous diseases, such as obesity, diabetes, hypertension, and I can keep going, we might have to take a different approach and look at the phenotype first and see how the phenotype of these complex diseases are driving our genome, our epigenome, transcriptome, proteome, and metabolome, and microbiome, and so on, in all the omics. So for that reason, another way of looking at and taking this slide from metabolome is to look at the genomics, that what come from mom and dad, and then our environment. Our environment is what we do, how much we exercise, what diet we eat, what medications we take, what's our microbiota, and all the things in our life that has affected our genome, and both together give us our phenotype. So when we talk about obesity, we try to find out what is the obesity phenotypes. And the obesity phenotypes, again, I'm going to simplify it as much as I can, is a disease of energy balance, and the phenotypes are driven by intake and expenditure. And intake is hunger, satiation, satiety, and emotional eating. And expenditure is the resident expenditure, our physical activity, which is non-exercise physical activity, and our exercise. And when we lose this balance, we start storing fat. And the storage of fat comes with two problems: adiposity toxicity and the excess of adiposity. So that actually tells us that we had drive to insulin resistance, inflammation, metabolic syndrome, cancer, joint disease, sleep apnea, depression, and GERD. So studying all these phenotypes in obesity, after studying 600 patients and using machine learning tools, we came up with what we think is the 4 main phenotypes of obesity. There might be more, but these are what we think are the most important ones. First, we have the hungry brain, which is abnormal satiation. We don't feel full and we just keep eating. Then we have hungry gut. We come to a meal, we feel full, normal, but then within an hour or two, we feel hungry again. So that's what we call abnormal satiety. Then we have emotional hunger. People are eating for their emotions, either positive or negative rewards. And then finally, we have our slow burn, which is our basic metabolic rate, or metabolism is not working appropriately and we're not burning enough calories. So these are our key obesity phenotypes. And these obesity phenotypes were identified using machine learning. And more than that, it has actually helped us drive and find a phenotype-guided intervention in which each of these phenotypes will have their own lifestyle intervention, medication, devices, and surgery. And I'm putting here the references of multiple randomized placebo-controlled trials, which we have validated these interventions in a phenotype-guided intervention, which brings precision medicine for obesity. However, today, I'm not going to keep focusing on this. I'm going to share with you what we're doing on the next omics level. So now that we know that obesity can be subclassified in phenotypes and we know these key phenotypes, how can we use omics to try to understand the next level of what's going with obesity? And in order to do that, we want to understand the pathophysiology of hungry gut. And myself as a gastroenterologist, I really want to understand why we have this abnormal satiety. And in order to do that, we want to rely on transcriptomics. Transcriptomics are great. We basically see how the DNA is converted into RNA, and that RNA might convert into a protein. So we want to study these enteroendocrine cells in our gastrointestinal tract called enteroendocrine L cells. They're about 1% of the cells over the mucosa of the gut. There are 10 types of them, which produce hormones that secrete into the content after we eat or after exposure to the environment, such as microbiome. And then we regulate the digestion, the motility, and food intake. But the problem when you only have 1% of the cells is that the colonic mucosa, the mucosa in our gut and in a small bowel is quite heterogeneous on itself. So using a whole omics technique, using a whole transcriptomic technique, it's very limited because we study all the cells at the same time. So in order to answer that question, we actually did a next step of omics, which is single cell transcriptomics. We took biopsies of patients with and without obesity, and we phenotyped those individuals, and then we did a single cell transcriptomic or single cell RNA sequence to study the transcriptomic profile of patients with obesity, acknowledging that there's a lot of heterogeneity in the mucosa itself. So doing that single cell analysis, we're using transcriptomics, we're able to identify 20 types of cells that are unique in our gut, and we identify our cells of interest that I'm circling here with this circle here in red. So there are many different cells types in the gut. So we're breaking the heterogeneity of the mucosa level using a single cell type, using single cell transcriptomics. And then when we look and combine the single cell transcriptomic tool with a phenotype tool, we can see that if we look into all obesity compared to patients without obesity, so health, we can see in this volcano plot that there are genes that are going to be overexpressed here to the right or downregulated here to the left. However, when we look at patients with or without obesity, so all with obesity, but with or without the hungry gut, the abnormal satiety, we can see that there are other genes that might be overexpressed or downexpressed. So when we actually quantify these things, you can see in this figure here and see, if we would just look at lean or healthy versus obesity, there'll be a total of about 200 genes that were abnormal. But when we actually look at the obesity subtypes or obesity phenotypes, and we use that is breaking the heterogeneity of a complex disease, and then we bring a single cell technique to break up the heterogeneity of the gut mucosa, we can actually identify 2 times more genes that are abnormal as we have identified, and this the information is under review. So with that said, I just show a very small example of how we can go beyond DNA and omics and use omics as the next frontier. So when we talk about chronic diseases, the one treatment fits all is not working, and we need to move beyond that. Obesity is a complex disease with many different phenotypes, and we identified 4 key of them. Phenotypes may guide obesity interventions, and we have significant amount of data published to drive this intervention and actually help our patients lose more weight. And then obesity phenotypes enhance the understanding of underlying pathophysiology when using omics tools, as I just showed with example with single cell transcriptomics. So I'll stop here. Of course, I can talk about an hour or more about these things that I just briefly touch bases, but I will give enough time for my colleague to present as well about the next frontier of omics, as well as then have enough time for Q&A. So I can finish by thanking everyone who is actually doing the work, everyone in my team, my mentors, Dr. Camilleri and Dr. LaRusso, all our collaborators at Mayo and external. Of course, Sim, who has been very graciously funding of my research and our program, and then the NIH as well for supporting our research program. So I'll stop here, and then we'll have some time for questions after this. Thank you.
I am on my seat, excited to hear more. Dr. Antonio, the floor is yours.
All right, so I'll just pick up where Dr. Acosta left off, and that's, you know, thinking about other types of DNA and other types of multiomics that are happening in our body and other chronic conditions such as cancer. And just like Dr. Acosta, I have some disclosures to make. Just we do have a patent that is related to some of the content I'll mention, and I am a member of the scientific advisory for Leukobiologics, but that does not interfere with any of the content that I will talk about here. So my background is really as an astrobiologist. I really spent some time looking for a life elsewhere, and it's something I still do, and try to understand life on Earth and extreme environments. And throughout that journey, what I really learned is that no matter where you go on Earth, you find life. You really go to the worst places on Earth to prove that, yep, life is not here, but it always is. And so that really, you know, should give people pause when microbes can, you know, survive pretty well on Mars analogs, and also in even Titan, which is one of the Saturn moon's analogs. Oh, sorry, this is, and under several layers of ice, as you see in the microbiolites there. And I have this little graph that you can see on the bottom here, which is to kind of bring awareness to people who might think that microbes are too small to really impact human health. This is what they have done to our planet, which is you know, much bigger than we are. And you can see this great oxygenation event. I don't know if you can see my mouse, but right here you see the great oxygenation event, and that's was entirely produced by bacteria. And that really allowed us to come into existence after this warring billion of things not happening, much things happening, and then multicellularity being developed. So the microbes did this to our planet. So if they have this kind of impact at those types of dimensions, imagine what kind of impact they can have in our body, of course. And not only that, this is something that we also were involved in sending microbes to space and to the International Space Station and understanding how they survive there almost 2 years under space conditions, and they came back alive. You can see how they changed. This is the ones that stayed on Earth in similar conditions, the ones that were kept inside the space station, and this is outside the space station, and you don't need any omics to understand that something changed, but they survived. And now you use the omics and single cell technologies to really understand what exactly changed and what does that mean for our journeys when we start planning long-term missions to Mars and those sorts of things, and understanding life limits and what that means to our health as well. Because when we go to space, it's not just us that are being impacted, it's also our microbiome and everything that goes with it. This is something I really like that talks about the concept of holobiont, which is how microbes are really integrated with our genome and all the omics that come with it. And these are particularly interesting to me, the blue ones that are the co-evolved ones. So these are bacteria that basically our body has evolved to expect to find there, to be there, to perform certain functions. And if it's not there, it becomes a problem. So people usually think of microbes as a pathogen, but not having the good microbes that perform necessary functions can be just as detrimental, and that's one thing to keep in mind. And just like plants, they have evolved to depend on this nitrogen fixing bacterial species that fix nitrogen for them. Otherwise, they can't really survive in the soil. Our microbiome also has evolved to depend on some of these functions. When we eat fiber, this is pretty much we're feeding the microbes, not ourselves, and that's important to our body functions as well. So this is something to keep in mind. So when we start thinking about what does this all mean to cancer, I'll touch base on endometrial cancer. This is the system that I know best. And this is a cancer that, although it's not much talked about, I think it is actually quite a significant one. It is more common than leukemia, and it does kill more people every year than melanoma, which is remarkable. It also, the incidence rate is expected to pass that of colon cancer by 2030 if nothing is done to change that. The current approaches that we have lean a lot on symptomatology, which works okay for, I'd say, Caucasian women, but for African-American women is really a big deal. And because the symptomatology is different, the phenotype, as Dr. Acosta was talking about, can be different, and it's not serving that population well, and that's something for everybody to become aware. So we set up a study, and you'll see a lot of faces up in my slides, just so you know what to blame them for, and all the good stuff, of course, but this is the part that they really contributed immensely. So we set up the first study to characterize the microbiome in endometrial cancer, and it was a very long and strong effort between different departments, surgery and pathology, and to do all these collections, and several papers resulted from that effort. And one of the things that we found was that there was indeed a different microbiome that characterized patients that had endometrial cancer versus some other benign condition, and you could distinguish them. You also see that there's a particular, what we call the endometrial cancer microbiome, that is increasingly getting worse throughout hyperplasia, which is a precancer. Type 1, which is the most common. Type 2 is the most aggressive, difficult to detect, and you can see some kind of move in that direction of the microbe presence. And then you can see sort of comorbidities, how that changes the environment. You can see the menopause has an impact in the microbiome. BMI in the reproductive system did not show actually significant impact, even though we know in the gut microbiome that's a very different story, for example. Vaginal pH also had a strong impact. And this is just an illustration of how all these things come together. It's a pretty complicated story. As you can see from these networks, you can see that these microbes do not exist in a vacuum, right? They interact with each other, they talk with each other and with our body. And there's the context matters. You can see by the thickness of these lines how much they're connected with each other and interact. So if we're concerned about this particular one, which we are, because of this thick line here, you see it's not that easy to get rid of it, because it's going to have a complex network that can bring it back up, that will sustain its continued development. And so we have to think about a systems type of level approach to really try to solve problems. And then another omics that we see often intermingled with microbiome is epigenetics and methylation in particular. In this case, we can see how patients, you can see how these are control patients that never develop endometrial cancer. These are patients cancer patients before they developed cancer and after they developed cancer, and you can see how the methylation markers keep increasing, this particular 4. And, of course, we think, knowing what has been out there in the literature, is that, the microbes involved in this, and that's something that we are investigating to determine if the 2 markers are basically connected or not. And they're trying to determine, as Dr. Acosta showed, those layers, how do those layers interact with each other and influence each other, right? And who's the chicken and who's the egg? That's still part of open questions. So studying the body as an ecosystem, this is kind of what we're trying to do in the lab, and trying to create different technologies. Some of these things sometimes require kind of different approaches because it's a different question that is being asked. This is what we call a drop-off system, is a droplet optofluidics merge. And you can see here how small it is. It's a really, really small microfluidics chips that we use. And then we can insert the cells basically in what we call the microcosmas. They're inside these little droplets, and you can see the cells inside the droplets. We can kind of move them and orient them in different places. We can use what's called as Janus particles, where we can compartmentalize and keep human cells apart from microbiome cells and compounds that we want to test the impact of, for example. So trying to recreate kind of a natural system as much as possible. I think of it a little bit like a cell farm, like you think of an ant farm, where you try to observe the system as much as possible and retain the complexity as much as possible. But just because all these things are very complicated, complex knowledge does not have to have complex applications. It can have simple applications, and this is also important to retain. This is something we've done with clinical fellows and research fellows in thinking of biomarkers to detect endometrial cancer. We have published this on a positive predictive value for the presence of this particular microbe of 0.86, meaning if you have this particular microbe in your vagina, you are in your postmenopausal, there's an 86% chance that you probably have endometrial cancer, basically. And we see that in all type 2 cases, which are very difficult to detect. We're also looking into using the microbiome as a predictor of recurrence in the disease. We do see some kind of separation. That seems to indicate potentially a role or just as a biomarker, but still interesting to know. And we are interested also in simplifying this as much as possible and put the power really in people's hands. This is a project that we have in cervical cancer, but that is really with a humanitarian kind of streak, trying to help the situation in the Democratic Republic of the Congo, where they really could use easy detection method, similar functionalities of pregnancy test. In essence, we developed these kinds of sticks that you can dip in urine to determine if you have a high-risk strain. And you can see that it basically turns blue if you do have it. If you don't have it, it doesn't, and it's kind of stable. And that's kind of the point that a patient could do this on their own and kind of know whether they need to go to the hospital or not. So these kinds of applications, so you take complex knowledge, try to simplify it into a clinical application, and furthermore, to try to really bring that to patients' hands as much as possible is what we try to do. So we're very much aligned. The needs of the patient come first. And I think Dr. Acosta really talked about that cancer treatment and chronic diseases often, fail to provide the intended outcome. And that's because we all know this, right? We know that there's one treatment for all, right? There's going to be people that are going to benefit from that, but others not so much, or it's going to be toxic, or it's going to be both toxic and not helpful. And we know that. And we just accept that because that's the current paradigm we have of treatment. And it works well for a lot of things, but chronic diseases is kind of an issue. And so I think of it as like Dr. Howe's this, idea that, as the patient will see you now, maybe a less mean version of Dr. Howe's, but kind of where the patient and Dr. Acosta talked about phenotype, but really having that as the center of care instead of the disease. And having a dedicated team to basically understand that particular phenotype or patient. And I think that, uncertainty and complexity is unpleasant for humans, I think inherently, and this is why we have been doing this for a long time. We see something like this and we're like, oh, I can't, we can't make sense of that. So we're just gonna say A goes to B through all of those loops, and then we're like, oh, the loops are probably not important. Let's just say A goes to B, and then we can just keep pulling these things out, right? So we have this urge to simplify, but the problem is that in this particular complex problems, that's their nature. They're complex, and you can't simplify them because the systems only functions in that manner. So if we study part of the problem, we really only get part of the answer, and that's my concern, is that maybe some of these answers are really in those loops, or how those loops interact with the loops of the other strings. And I see really a comparative example with the astrobiology world that I think about often as well, which is that complexity is uncomfortable and unpleasant, but it is real. And this is just an example of a Hubble Space Telescope image that is just a little sliver of the sky that is taken in extreme deep field. And you can see this image, and this is not stars, they're galaxies. All of what you're seeing here is galaxies, even the little dots. That's just because they're too far for you to see anything else. But there's about 5,500 galaxies in this field of view, and each galaxy has billions of stars, right? And the most distant ones are about 13 billion years away, light years away. And we're seeing them when they were about 500 million years old. That's just how, it takes for the light to get here, for us to see it. And Earth was formed about 4.5 billion years ago. So the conclusion of that is that we're basically presently looking at the universe billions of years before Earth even came into existence, right? And how do you how do you make sense of that, right? And this is kind of, you know, the contact Jodie Foster and then Hugh Gloria's Dr. Hausen, you know, character that, you know, where is life likely to be in this scenario and why, and where is cancer going to be and why? And I think we can try to simplify these questions as much as we can, but we have to retain the fact that complexity is real, that is the real system, and that we need to really understand that before we can simplify it in parts for applications. And I think that's the part that I want to, I guess, convey. So this is all kind of like brain teasers for discussion, and really looking forward for this.
Thank you, Dr. Antonio. You know, I majored in physics when I was in college, and astrology was my favorite thing, and now I'm just doing nephrology. So this is kind of a fun deja vu.
You can do both.
Right. All right. Well, while questions come in, I've got a few questions for both of you. We'll start with Dr. Acosta. So Dr. Antonio talked about the microbiome. You talked more about the phenotypes and the different ones that you targeted, and looking at cell-specific transcriptomes. And can you speak to the microbiome of obesity, specifically in that phenotype of interest that you shared with us today?
So I know a little bit about the microbiome, but I'm not the expert. So let's take this with a grain of salt. But I think the microbiome is one of the other extremely important omics in this whole interaction of our environment with our genetics, that both our genetics have this predisposition for obesity. We live in an environment that is predisposing us or pushing us to preserve energy and weight gain as we are less active and we eat more. And then our bacteria, especially our colonic microbiota and small intestine microbiota, has adapted to actually be a key player in this whole genetics environment interaction. Jeff Gordon in the initial key publication in Nature in 2006 already talked about the interaction of the microbiota with in twins, one with obesity and one without, that was transferred to a mouse. Significant progress has been made since then, and there's multiple different approaches and interventions that are currently under being tested in order to see if by modulating the microbiota, we can actually improve health, and not only do that, but also help with weight loss. However, it's not the only thing. And that, I think, is very important, because sometimes people might think, oh, the microbiota and the treatment of that. Sorry, Dr. Walther-Antonio.
No, I agree. I totally agree.
But it's a key component of this very complex puzzle. And it's one of the pieces of it, and it has to be taken into consideration. And especially if we talk about how our food has changed, how our activity has changed, and how our bacteria is also sensing those things.
So along those lines, can you gave us kind of a cliffhanger and you left us hanging. You didn't tell us the next step after looking at that specific cell, endocrine. I'm probably butcher the name there. So what's the next step? So you've identified signaling, you've identified there's variability. Where do you see this work going in the next 10 years?
So that is the beauty of what we are doing and bringing it to the next frontier. So now we can do, using systems biology, we can take and know the DNA of an individual. Now we can know what they're doing at the individual cellular level. And then at that cellular level, we can see what's the protein that is functioning and what is the abnormality. So when we talk about obesity, and many of our patients with obesity, about 30% of them have abnormal satiety, we will be able to know why exactly they have abnormal satiety. So hopefully this will be a matter of a future lecture that probably need a little more time to dig a little bit further. But we have already identified multiple different new targets or new proteins that we identified in the transcriptomic profiling. We have confirmed at the proteomic level, and now we're looking exactly at what they're doing at the metabolomic level. And those interacting with the microbiota, it might give us this phenotype of what we're calling hungry gut of abnormal satiety. So if I can daydream, in 5 or 10 years from now, we will have specific targets either with diet interventions, specific diets that will be targeted to our individuals, specific microbiota or specific medications that will help us treat this underlying condition of hungry gut in one of these new targets that we're moving forward. And then we need to do the same thing for other obesity phenotypes. Of course, doing a single cell analysis of hungry brain or emotional hunger might be more difficult because we'll need biopsies from our brain, different areas of the brain. But we need to find new tools there. And then there's this new omic, which is imaging, which imaging can be a lot of things. So we can have deep phenotyping with imaging like MRI, working with our colleagues, John Port. That will bring all to the next frontier. And then you name it, AI and all these other tools that we'll be using will help us identify what should be the end goal, help our patients by giving them the right intervention to the right patient.
Thank you. Thank you, Dr. Acosta. It's exciting. So Dr. Antonio, question for you. I was very intrigued to learn about that one association with that specific bacteria and how it's associated with such a high positive predictive rate for endometrial cancer. Do you foresee us at some point in the near future to be able to do a mass testing, large scale, kind of similar to what you've done with the HPV testing for the Republic of Congo, where you look at everybody and look at their microbiome, and based on their microbiome data, guide early treatment before the development of cancer?
That would be the dream. We do have, grants pending, I guess, basically on that idea, to test that idea. It involves a joint effort between Mayo and Johns Hopkins and Emory University that we are trying to enroll patients on all those sites to determine not just the microbiome, but also the epigenomic methylation aspect of that, because we really want to understand what is our timeline to try to stop something from happening, and what should we be stopping, and what comes first and next, and that really needs to be just tested to see whether that would be something viable. It wouldn't. You could offer this to all women, but I think the population we're focusing more is on women, particularly postmenopausal women who present with bleeding, and they typically get an endometrial biopsy to rule out cancer. But then we do know that, even if they have a benign finding, we know that 1 out of 4 women with endometrial cancer have had benign biopsies in the past. And so what we want is to really know who they are before they get to that point where they do develop endometrial cancer, and stop that flow of events as much as possible.
Don't have any other questions up, but are there comments or thoughts that you want to share with your audience to kind of get them excited about the microbiome, and or perhaps justify the role of the microbiome to those who may not be believers?
Yeah, so I had a question for Dr. Acosta. I don't know if you're familiar with Day 2 efforts. Are you familiar? Yeah. So I think we're very aligned in the way. I don't think the microbiome explains everything. All right. I think it is an important and crucial aspect of our health that we need to take into account when we think about what's happening, or treatment resistance, metabolic capacities, metabolizing drugs, and all that. I think it needs to be taken into account. And sometimes it might be a big role, sometimes it might be a small role. We don't know. But you were talking about knowing the phenotype and what how you determine then what to do about it. The Day 2 effort is kind of trying to design a personalized diet, right? Something that they would identify, oh, this is, to you, an apple is good, and maybe to, somebody else is going to be a bad thing. And should we do that sort of, what do you think about those types of efforts to personalize those types of diets, and maybe exercise regimens as well, and all that?
Yeah, absolutely. And it's a great question. Not only with the microbiome, there's multiple companies who are, and many scientists doing research in this space, including ourselves. And the idea is to test for different variables. We can do DNA testing, we can do microbiota testing, we can do metabolomics testing. People are working on a proteomic panels. That's the conflict of interest that we have at Mayo, that we have developed a multi-omics panel. We combine different things. But the key concept of this is that we will be able to identify using these variables what should be the specific diet of an individual, or even medications and surgery in the future. The company that you mentioned, which I think has a very nice technology called A2, which actually Mayo did the validation study in which it was able to find what a specific diet an individual should take and do in order to reduce their postprandial glucose, or their postprandial glycemia. As we know, many patients with diabetes, they have high levels of fasting glucose, but also when they eat, they have these very high levels of glucose to certain meals. That's what we call the glycemic index of food. But now we take it to the next level. We knew about the glycemic index of food that was discovered back in the 60s and 70s. And now we added the microbiota. And combined those two things, so a better phenotype and an omics tool, we can actually improve the prediction of what diet an individual should be. So they're great tools, they're advancing the field, and they're helping the purpose that is improve health and improve disease management. So I'm very excited about all these things. And what the key thing is that I think when we talk about, especially in the wellness space, is we need to learn to separate the noise from the data. We see a lot of new companies saying, oh, this is the DNA wellness profile, and follow my metabolomic pattern and you will lose weight. It's important to make sure that there's data behind it. And many of those studies have been only done in association studies, which certain people have certain gene or certain microbiota, and we think we need to change that. The benefit of companies or studies like Day2 is that they have actually done intervention studies to actually validate the data. The same thing we're doing at Mayo is taking it to the next level, bringing from an association to an intervention study in a randomized placebo-controlled trial. And that's the level of evidence we need to actually apply this to patients and help patients either with their diabetes or in this case with obesity. So I'll ask you a question back, which I think is very important when we talk about omics, Dr. Walther-Antonio, is one of the things that we constantly see is the heterogeneity among different gender and in this case, races as well. And then location matters a lot. So can you tell us if you have seen any difference when you do your microbiota studies here in Rochester, Minnesota versus in the Republic of Congo? And is that applicable in both places with different populations, or you have seen that it's the same studies or needs to be individualized even further?
So we have compared, mostly my group focuses mostly on reproductive health, not as much the gut, but there's a lot of literature on the gut. We have done studies, for example, where we compared a cohort that we had of women, pregnant women, for example, and the vaginal microbiome as it correlated to normal delivery or preterm delivery, for example, versus a cohort from Baltimore that was primarily African-American, ours was primarily Caucasian. And interesting things come out. So what we see, for example, is that when women are not pregnant, they seem to have a different microbiome that is distinguishable between Caucasian population, African-American in this case in study. But once they are pregnant and during pregnancy, it's kind of the great equalizer. They all go to the same place in a healthy pregnancy. And if they don't, that tends to indicate some issue pending to occur. And we do see those in preterm rupture membranes and that sort of thing. So it's complicated, I guess. And it's probably influenced a lot by diet and the gut for sure. We do see populations that move from other places into this country that, we do see them changing and starting to develop the diseases that are more common in this country than their country of origin. And that it seems to be heavily influenced by the changes in diets and environmental exposure. So I think it's hard to tell, those differences are, there's probably some component that is genetic, some component that is environmental, and it's some combination of both that results in some of the differences we see. But when we're applying applications, it's really important to know because, for example, even in the Democratic Republic of the Congo, the HPV strains that are high risk are not the same, the prevalence is not the same that we see here, for example, so you have to focus on different strains that are the most relevant there. And when you come here, you apply to that population what's relevant to this population. So I think it's important to be aware of those differences and utilize them to improve patient care in those areas specifically.
I see there's a question.
Yeah.
So I'm going to read the question. Using omics to treat obesity is fine. However, is anything being done to use omics to prevent obesity, identify susceptible groups? Absolutely. There is a very large study came from the Harvard group. The Lee Kaplan was one of the key authors, in which they identify a polygenic risk score. So using around 2,000 genes, which will tell us from birth who will gain the most amount of weight in the current environment that we live that is obesogenic. So that will tell us who will gain weight. What it's not telling us is how they will gain weight. And that I think is the next frontier, because basically every human is susceptible to obesity because of the environment that we live. And every human that has come to today, because since we were gathering hunters or hunting gatherings and we needed to survive, we have created a lot of pathways, genetic pathways and metabolic pathways to be able to preserve energy. So we are machines, we as humans and most of animals that are quite good at preserving energy. I like to explain to my patients that for every 100 pathways, genetic pathways that we have to store energy, we have only one to burn energy. So when we have that problem, and now we live in abundance and we have a lot of resources and we eat a lot more and we exercise and we're less active, we are all susceptible to gain weight. But the question that I think omics needs to help us, and we already have an initial blurb from this polygenic risk score, is telling us how we're going to gain weight and what is going to be the mechanism. Because let's just separate the two big equations of obesity. If we know that someone has more predisposition to gain weight by having a slow metabolism, that individual definitely needs to be more active and hit the gym. But if we know that we're going to be someone who feels more hungry, doesn't feel full, and has more predisposition for emotional eating, maybe we don't need to make such an emphasis on the exercise. We need to make more emphasis in the appropriate diet to prevent them not feeling full and feeling hungry all the time or looking for emotional eating behavior. So I think that's where the field needs to take us is, of course, we have 40% of obesity currently in the country and we need to address that, but we need to prevent for the other 60% to follow the same path. And I know everyone, and I will say absolutely everyone, I haven't met a single person who doesn't care about their weight. And we're all thinking about that because it's part of our health. And we want to know what's the right plan for me. Do I need to hit the gym or I need to watch what I eat? Or maybe both. I think I would love to get there, and hopefully with the research that we're doing and all the help of all the participants who come to participate in our trials and all the funding sources, we will get to that big question is, how do we stay healthy? So what's health, what's healthy, and how we stay there and prevent disease. So that's, I think, where really we'll be able to, to really change mankind if we can prevent disease and prevent obesity and understand who to do what.
Just a follow-up question on that. So I'm very fascinated by the longitudinal and as time, the time factor into this. And do you expect this to be time sensitive in the sense that you expect it to change through time? Say, for example, there was a person who had, weight gain that was due to lack of activity, say, but then, 20 years later, they're having the same issue, but now they had some kind of traumatic events that might have caused emotional eating now this time. Is that something that would have to be reevaluated every time to determine what changed? And or do you expect it to change or to be fairly stable, I guess, is the question.
No, absolutely. And that's unfortunately the complexity of the phenotyping, that it's an ongoing evolving process. We have our genetic predisposition. For example, a person, about 1% of patients with obesity, with severe obesity, will have a rare genetic disorder of obesity that drives them to eat more, so hyperphagia. That's how they're born and that's how they have. But if they go through a traumatic event, using your same example, in the teenager years, And that induces a significant amount of stress, and that stress drives them to depression or anxiety, that then drives to emotional eating. Now we have a problem with a hungry brain with emotional eating. And then let's say that we go in and we address this problem, we might be able to help them with their hungry brain and the lack of sensation of fullness, maybe with a medication. And then we might need to do therapy to address the emotional eating and combination of those. But that same individual then will get to, let's say, the 50s or 60s and be postmenopausal or postandropausal. And then their metabolism is not so efficient. So testosterone has come down, estrogen has come down, and then maybe the phenotype that they might be having now is an abnormal metabolism. Or, let's use an example, they have a severe thyroid problem, or they're taking a medication that is shutting down their metabolism because of any other problem, and then they have another phenotype. So that's a problem with phenotyping that is not static. It's not like our, as I was using as an example, it's not the cooking book, the recipe. It changes all the time. So the chocolate that I was going to buy to do my chocolate cake now is not coming to the store, and I need to find a new chocolate that might change my recipe. So I don't have milk chocolate, now I have dark chocolate, so my cake is going to taste different. And it's the same thing with phenotypes. It's constantly evolving. You know, we get a round of antibiotics because we get strep throat. My phenotype will just change because my microbiota changed, right? We go into a pandemic like COVID, like we're right now. We're all under stress. We're all living in this. We're working from home. We're actually working from the kitchen. So likely we have a combination of a new phenotype that is COVID-induced emotional eating by working on the kitchen, and we're getting it. So this is constantly evolving, and we need to be aware of this and change it.
Well, you're making us all very hungry, this chocolate cake.
Yes, absolutely. I think there's another comment. I will read it here. Great information. I look forward to future changes when you achieve results of the intervention studies and the role of microbiome diets in managing obesity. So thank you for the comment, Jody. And it's a great question, comment. I also would like to invite everyone at Mayo. We are applying many of these tools. For example, Dr. Walther-Antonio mentioned about one of these microbiota tools to help us with postprandial glycemia. That is something we have at Mayo. We're trying to make sure that it moves from research to a clinic. Many of the things we have done, we have moved them to the clinic as well, because these are non-invasive tests that actually help us with the management. And we have tons of studies. So if anyone wants to participate in our studies, everyone is welcome to come and contribute with their time. Most of our participants benefit from weight loss, which everyone wants to do. And then we all together, we all as a community, we all as humans, and all of us suffering from obesity, we all benefit from learning more. So I'll invite everyone to participate in the studies. And there's tons of things going on at Mayo. And if you're not in Rochester or in one of the other Mayo sites, there's always things to contribute and collaborate and participate in order to understand these more and really bring omics as the next frontier to the present and understand how we can advance science.
I don't think I could have ended it as well as you have, Dr. Acosta. Dr. Antonio, do you have any lasting remarks?
Thank you.
No, I think our discussion is really to try to bring awareness to people that things are complicated, and we need to sometimes we need to take all of it into account, and it changes through time, and it's a hard job for doctors, and I don't envy them trying to figure out in all of this and trying to come up with a solution for patients, and we're all working hard to make that a good decision making for everybody. So
absolutely.
Thank you both. It was really a rich discussion. I learned a lot and excited to invite both of you back to give us a follow-up plan. So thank you all for participating with us, and we look forward to seeing you for our last webinar, hopefully not last, but October 1st, the last scheduled one, and then our virtual conference, October 14th. We encourage you to register and join us for that conference as well. Thank you and have a great day.